This is really a powerful program, and we're so glad that you can join us. It really focuses on how we treat cancer. And when someone receives a diagnosis of cancer, it is just frightening. We all know someone who has battled cancer sometime during their lifetime, and it's important to recognize that nearly half of us will receive a cancer diagnosis during our lifetimes. Cancer is the second most common cause of death in the United States, killing 500,000 people of all ages nationwide each year. So how has cancer treatment changed or improved over the years? Is your prognosis better than someone with a similar prognosis just that in the State of the Union address in January, President Obama announced a new national effort to cure cancer, citing the 2016 budget increase for the National Institutes of Health. He was going to add and recommended up to $2 billion be added to the already $32 billion for the NIH. And this is the first uptake in a decade that science research funding has received. So this program will outline the history of cancer treatment and outcomes. It will focus on the best current practices from experts in the field. And it will preview what is on the horizon for the next generation of cancer treatments. And this is such a special privilege for me to introduce two individuals whom I have the greatest respect for. They are just passionate about their work in cancer and helping patients and coming up with new and novel therapies for cancer. I'll introduce both of them, and then our first speaker will be Dr. Giles, followed by Dr. Davidsen, followed by questions from the audience. Dr. Francis Giles, known as Frank Giles, is professor and chief of the Division of Hematology and Oncology within Northwestern University's Feinberg School of Medicine. He is also deputy director of the NCI-designated Robert H. Lurie Comprehensive Cancer Center of Northwestern and director of the Northwestern Medicine Developmental Therapeutics Institute. And I would also pointing out to you this evening that Dr. Leon Platanius is with us, also over here, and he serves as director of the Lurie Cancer Center. I would just like to say that Dr. Giles has really committed to making sure that pediatric patients receive the similar kinds of opportunities to engage in cancer clinical trials as adults. And this has not been the case in our industry. But he is really making a conscientious effort to work very closely with pharmaceutical companies such as AbbVie to make sure that this happens. Our second speaker will be Dr. Steve Davidsen. He is Vice President of Oncology Discovery in AbbVie's Research and Development Organization, where he is responsible for discovery and research efforts of AbbVie's oncology programs. Prior to AbbVie's separation from Abbott, and it was amicable, okay? They just kind of split off because they had two different areas to focus on. Dr. Davidsen served as Divisional Vice President of Cancer Research. Dr. Davidsen first joined the company in 1986 as a research scientist, and I would just say from our really personal interactions with Dr. Davidsen and his team, we have learned so much about how industry focuses on meeting milestones and thinking about new and novel therapies. It's really been a privilege to work with him and his colleagues. So again, our first speaker will be Dr. Giles, and we will hand the podium over to him. Thank you.
Thank you very much. So, good evening, and thank you very much for that. Mary, as you all know, is not alone a brilliant scientist, but excessively generous and gracious. Your home university is very lucky to get you back. Congratulations. It's a great pleasure to be here. It's a great pleasure, and I'd like to thank Leon for joining us. He's the director of our cancer center. A lot of other things to do, a lot of other places he could be, and It's a great privilege. One of the things he told me when he was chatting with Steve and I, I'm sure he won't mind me sharing this with the whole world, is that his morning routine is to go on a treadmill and watch horror movies. However, he made an exception this morning and watched Steve and I's segment on TV as a substitute for Slasher 17. And I'm still trying to debate what to read into that substitution. But Leon, thank you. Thank you, Leon, I think. So Mary used, and it's clear from her demeanor, that something unusual is going on, and that is we're perfectly prepared to come in front of our peers, students, everybody, take up their precious time and express enormous optimism about what's going on in cancer. That in itself is somewhat unusual. What I'd like to share with you all very briefly is what exactly has led to that optimism. So very fundamentally, a cancer cell is crazy. What do I mean by that? It's biologically illogical. It will replicate, multiply, spread, will move into organs where it has no function, will steal nutrition where the host, us, you or I, can perish. It behaves completely illogically. That's always been known. That's why it's always being so feared. And as Mary said, still is responsible for so many deaths per year. But the critical issue is it behaves in a crazy, aberrant, illogical manner. That's the first big point. The second is cancer should not be allowed to gain traction in our bodies in the first place. In the womb, we already are starting to evolve a very sophisticated immune self versus non-self recognition system, as you all know. Our immune system evolved to protect us from environmental threats, infectious agents, etc. And it evolved a long, long time ago quite sufficiently to recognize cancer cells, most of which have totally abnormal proteins and other excitants, if you wish, of T cells or immune cells, and yet the cancers develop. By first principles, the only way that should happen is the immune system is exhausted, distracted, or most importantly, as Steve will go into in great detail, negotiated with. The cells that are capable and perfectly capable of eliminating the cancer are told either I'm not here, I am here but ignore me. Some way they're negotiated with. And these are not ephemeral concepts. What we now know in both cases, both the crazy behavior and the fact that the immune system allows this to happen, are physical, chemical realities. Mediated in one case by abnormal genetic behavior, and in the others, we think predominantly by a series of exchanges between the cancer and immune cells saying, leave me alone. What's the point? The point is that both codes have not alone been cracked, they're increasingly cracked. And as the code has been cracked, actionable, vulnerable points, either in the genetic code in terms of a target or in terms of the conversation between cancer and the immune system, that has led to the flood of approvals that you'll have seen in the last few years of so-called immune modulatory drugs. These are two parallel revolutions. We're learning the code. All of science is contributing to learning it, quantifying it, analyzing it, defining actionable targets, and doing something about it. And the objective evidence, as you've seen, are these approvals, are the people who are alive, have prolongation of life, especially with tumors like advanced melanoma, subsets of lung cancer. Cancer is really where we really were quite desperate, and where the baseline were cytotoxic drugs that owe their marvelous heritage to nitrogen mustard gas. A straight lineage, a straight rationale, I'm going to give you something that kills cells, and hopefully it will kill more cancer than normal cells. That was the fundamental rationale behind chemotherapy for the decades we've had it. It's got considerably more refined than that. It still is a cornerstone of our therapy. But you can imagine what it's like to watch patients receive a targeted therapy that tells the immune system, get busy and do your job, as distinct from giving a drug whose only job is hopefully you'll kill more cancer cells than normal cells. These are multiple, parallel, realistic revolutions. So what's the basis for this? Well, the basis is all of science. And if I just, because Steve's going to, I think, really very elegantly focus on the immune modulation, so I'll only touch on that briefly. I want to touch more on the genetic targets, the genetic code, what drives the crazy behavior and what can you do about it. And here, as a general science audience, I think this speaks for itself. And it's basically the sequence, the cost, the time, and the timeline to deliver actionable information from the original Human Genome Project to what we now take for granted. And you see 13 years down to a few days, 2.7 billion to $4,000 and dropping. And for all of the laws that had been developed, as you know, for the increment in computer processors and their speed and their cost, those laws of increment have been totally blown away by our ability to analyze the genome. That's one of the key drivers of this revolution. However, it's also a time of, I think most of us would say that we've a sense we're at the end of the beginning. But quite how much further we have to go to optimize taking advantage of this revolution is not yet clear. Why do we say that? Because there are multiple layers. You may have an abnormal code. The gene shouldn't be there, and I'll give you an example. But then the gene has to make an abnormal product. That abnormal product can be destroyed, enhanced, manipulated. So there are many different layers to these aberrant instructions, but now the science, the computing power, the global collaboration is cracking those codes, but we do know there are 3 or 4 more levels. For example, epigenetics is something in the Lurie Cancer Centre we're extremely committed to understanding, analyzing, and turning into new therapies. We don't quite know the depth of the iceberg yet to go in terms of molecular aberrations, but we are beginning to get a grasp. And most importantly, the bit you see represented in the cartoon as visible is already translating into prolonged survival for patients. This lady fascinates me. In the west of Ireland, if you tell people you shouldn't smoke, they have the anecdote about Granny down the road who has never done anything right, exactly like Granny here. She's 100, this is not her first cigarette, and it certainly will not be her last. And these people always fascinated me, and the implication is obvious. This lady has something special about her ability to repair DNA damage. Maybe her immune system is absolutely superb at picking up cells that tobacco and its derivatives happen to drive crazy, but there's something special about her. The reason I wanted to share this is, again, not to really go into the immune modulation. Steve will do that, but you know, for many, many years when we've done these early phase studies, there was always something very frustrating. And that was every so often a drug or an approach that was deemed not worthy of further exploration. There was always that one patient, the one patient who did extraordinarily well on it. And all you were left with was an anecdote, you know, a description. We gave it to 10 people or 20 people. One of them did phenomenally well. The point is, people like Granny enjoying her cigarette are the one person in 10 or 20 or 30 who respond to a new therapy when everybody else doesn't. They're precious. You need to know what's different about either their disease or their interaction with the drug that makes them special. And that, as I'll show you, is why we're increasingly carving up cancer into subsets that we can actually do something about. Now, most of what we're talking about is, and it's such a privilege to share this conversation with Steve, it's all about collaboration. The silos that used to exist were not particularly constructive silos. And this is an idea of Leon's, and looks on the face of it like a straightforward idea, which it is, until you begin to see the enormous benefits. And specifically, we're very interested in the Lurie in giving people very concrete incentives to work together. If you're in a science lab and you're interested in some esoteric enzyme or some esoteric pathway, quite frankly, it might be in a zebrafish and might be considered by many to be esoteric. The fact of the matter is that if you've got clinicians who are seeing real people every day for whom they're desperate to find new therapies, sooner or later it's amazing if you have enough contact how one finds the other, how one finds a way to ask a question. And in fact, if you look at it the other way, if you look at some of the drugs that I'll briefly touch upon that truly have been transformative, and you leave all egos aside and go back and say, well, who gave us this drug? If you go far enough back, you always find somebody described as a basic scientist. And quite frankly, at the time, cancer was nowhere on their radar. except as Mary says, in their personal life, where statistically it probably was, but not driving their science. And what you have in a comprehensive cancer center like ours is the day-to-day communication where those bridges, are bridged by protoplasm, by investment, or by collaboration. And that is the collaboration with pharma, with AbbVie, with each other across the disciplines. Quite frankly, now, in any given day, you could be talking to a biostatistician, somebody who will tell us how do you keep lots and lots of patient information safely in the cloud and yet analyze the information you want. It is astonishing over the last 10 years how trying to increase the number of patients we cure with cancer has become a pan-scientific endeavor, and everybody's conscious of it. It's not an esoteric concept anymore. So what has that brought us? It's brought us to the recognition that we have to start looking at people as individuals. Their cancers quite often have unique drivers, and their immune system and the interaction with that cancer may well be individual. On the other hand, you have to find enough commonality, both medically and frankly commercially, to come up with viable therapies. And I want to show you how the science is assisting in that. The other thing is this is all intensely disruptive. As you can imagine, if you've been some decades in a discipline and you regard yourself as, I am a lung cancer doctor, that's what I do, let's say. And then somebody comes along and very calmly says, well, I've got news for you, there's no such thing as lung cancer. That's kind of different. Next month, for example, in the Lurie, we're going to launch a clinic which is one of the first, if not the first of its kind. And that is where people will come in and they'll actually be assigned and seen a priori from the second they arrive, not based on the histology, not based on whichever organ we happen to find the cancer, but on what we've identified as the driver of the cancer. And that may mean that a gentleman with advanced prostate cancer may be sitting next to a lady with advanced breast cancer, and the two of them may have far more in common than would ever hitherto have been dreamed of. And that's the way it's going. But can you imagine, I'm sure you all can, how disruptive that is in terms of our current educational system, the way our hospitals are structured, the way our clinics are structured. So you have a confluence of science here at a very interesting time. And it fascinates me that disruptive has been made a negative verb. Actually, this is very, very overdue positive disruption. So let me give you a very, very short vignette that's very Chicago pertinent. Janet Rowley, who died recently, was a colleague in the University of Chicago. In the early 1970s, she showed the fact that what had been identified in 1959 specifically as a small chromosome in a group of patients with leukemia wasn't actually a small chromosome, it was part of a translocation, the 922 translocation. When the chromosomes translocated, they wrote a new bit of code that should not be there. That code generated a new protein, and that protein drove this curve. So what are we looking at? I remember this curve very well for a very selfish reason, and that is in Irish medical school exams, you always got extra points for a drawing. And we're not all artistically gifted. So I was always hoping that CML would come up with a question because this is a very easy curve to draw. And I remember many years later when we were allowed on the wards meeting the first CML patient feeling really horrible. The reason it was an easy curve to draw is very simply 15% of people came out of a kind of quiet looking phase and died in the first year. And then every year, 25% of whoever was left died until nobody was left. That was chronic myeloid leukemia, the first human malignancy in which an identified specific chromosomal abnormality was identified. And in this city, Janet and her colleagues figured out it's not just a loss of a chromosome. They've had an exchange, they've written a new code. That's what drives this disease. The new code put an accelerant next to an enzyme and basically said, your job normally, Abel, is to drive a certain degree of controlled cell proliferation. Let's see what happens when I put an accelerator next to you. What happens? A lethal disorder that kills on the curve that I showed you. But now we've gone from a chromosomal abnormality to what we call a target, because Abel is druggable. In the meantime, while we were evolving towards these new very targeted drugs, a drug or a class of drugs called interferons came along, and they moved the curve to the right, as we say. But look at the red curve. If you're on that red curve, statistically, you're still going to die of the disease. It's just going to take longer. But the shape of it speaks for itself. And then, I guess, okay, can I go back for a sec? Okay, and now let's get this back on track. Okay, now look at this curve. That's quite easy to draw. It's almost a flat line. It's what's called a normal survival pattern. In other words, we haven't quite cracked immortality yet, but these people are not going to die of their CML. So what's happening here? Now, these curves, when you look at those slides, they're actually this rare example of presenting data that's much, much better than it looks. You know why? Because on the interferon curves, young people went on. People who were physiologically and fiscally privileged enough to get interferon. They would have been 20 years on average younger than the average number or type of patients presenting with this leukemia. You know what's different about this curve? These are precisely who get CML, and this is precisely what happens to them. When we started treating this disorder in a very targeted way with interferons, there were long conversations. Basically, the conversation went, you could have interferon and this will happen. If you're very lucky, you might have transplant and this will happen. But statistically, you're going to die from this disease. That was the conversation. You know what the longest conversation we have with these patients now is about? Pregnancy. When would you like to have your child? How long do you need to be off the drug and you can get on with it? In other words, if you have a normal lifespan, why wouldn't you have a full life? Those curves have changed because of the transfer of science into the clinic. It gets much better than that. Imatinib is the drug that mediated that revolution. Weren't happy with imatinib. You get with computer scientists and chemists, and they say, could we make a better imatinib? The answer is yes, we can. Were they approved? Yes, they were. What's the point in the new drugs? Now the studies that we're running around the world go as follows. We'll give you 2 or 3 years of this drug, and we hope to be able to stop the drug forever. We hope you will not need any drug at all. Done. Finished. Get on with your life. That's the aim of the new studies, and all of that has happened within our lifetime. So that's the good news. Now let's take another angle in CML, and that is where it's got to a degree of genetic chaos that we can no longer cope with it. These curves are not in years. At the bottom there are months. The patients who had blast crisis 10 years ago still die at the same rate today as they did 10 years ago. In other words, beyond a certain degree of genetic chaos, we're not able to catch up. This is still the driver, but now the driver has recruited a lot of other drivers. But however, instead of being bizarre circuit diagrams, these are all new targets. Every one of those X's, we have specific drugs in the clinic going after those targets to try and layer on protection against what you saw earlier. So how, in practice, in Chicago, in the Lurie, what are we actually doing about this? Because here you have an obvious confluence of two desires. You want to set new therapies for our grandchildren, but you have people right in front of you who need an improvement today. And you have to balance that delivery. Like, it's all very well. Patient with cancer we'll see tomorrow, yeah, sure, they want to contribute to, society's good and something 100 years from now. They're actually quite interested in having something done for them too. That's kind of the point. So this OncoSet program, if you like, the sequence is get the information. The information is changing rapidly. Get it from as many sources as you can. Try and have as many interrogatory techniques. Add up the data as best you can. Evaluate. Well, we're not the only ones. There are data banks online. There are data banks all over the world. Pharma give us new drugs that are investigational, but maybe cognate agents we can match against the data. So we have a molecular tumor board where we take that data together and say, well, that's all very well, but what are we going to do about it? And then you have the bottom, which is the bit that I'm personally the most passionate about, and that is, well, the bit where you do something about it. Where you have a target and you go out and you search for agents and you try and maintain that pipeline constantly and try and keep it updated so that the sequence leads to the evaluation, leads to a potential target, and you try and do something about it for individual patients. Now, what does that look like in practice? Well, this is lung cancer, or a representation of it, and why I said there is no such thing. Because each one of these, we already have clinical proof. If you do something about that specific target, you do something about the disease. And they're mutually exclusive in most cases. So just to simply look at it and say, well, it looks like a lung cancer cell, that's the end of it, is clearly redundant. And as you can see in the cartoon, basically, The therapy will only work when you've got an aberration to market with. Now we have both the code and the drugs. So that leads to an entire change in the way we do things. First of all, umbrellas and baskets. So you take lung cancer, you say, we need 20 drugs to go after, you know, our group of patients suffering with lung cancer. Let's get our 20 drugs in, one way or the other, and have that portfolio. So you literally map, target, and try and ensure that you have agents to go after every one of them. And then there's the opposite. What happens if you've got an aberration, but it's actually relatively independent of what you or I would perceive as a cancer? So here's a very common one we have a lot of drugs to go after, but look at the percentages. It's spread across endometrial, breast, colon, ovarian, lung, head and neck, and pancreatic cancer. So again, you'll see the point of the Oncoset clinic. These two patients, who may look like they have two utterly diverse or different cancers, may actually have the one thing in common that makes it possible to improve their therapy. But that's a new way of looking at it. This is a very common target. You've got 4 things that can turn it on. You've got 4 receptors and 22 possible ligands. And they play with each other any way they wish across a spectrum of malignancy. You have to maintain a portfolio to go after that in every histology that you can. So very simply, what's changed, and one of the most gratifying changes of all, this is old drug development. I have a drug. I'm going to give it to everybody and let's see what happens. This is constructive drug development. Each patient is different. There may well be an overlap between what's driving, so it's sensible to give both of these patients drug C, but it's an utter waste of time to give them the others. So in other words, you're inverting the whole model. Don't worry about the drug. The drug will get approved if you generate reasonable data. Worry about the patient, worry about the targets, and design the whole thing around those individual patients. This is one of the most eloquent summaries written, as you might anticipate, by a professor of English. This professor came to Chicago for phase 1 therapies for his pancreatic cancer and ultimately perished from his pancreatic cancer. But I think his statements are pretty eloquent and pretty to the point. Our patients don't necessarily anticipate that they as individuals will be cured. That would be nice. They do want realistic. What can you do for me? What can I realistically expect? What am I facing? And they do quite incredibly bravely, day by day, contribute to the overall body of knowledge. We have a very deliberate sequence at Northwestern of doing this. Chemistry, physics, the modeling, the radiology. As Mary said, it is critically important to us. If you have a molecular lesion in a tumor, does it really matter if you're 6 or 86? So to do the current sequence, we've so much of a legacy of old drugs and old ways that we can't shed it fast enough. But I wanted to close on something that really is a way of introducing Steve and to say thanks for a lot of the collaborations that are going on, because the theme of the evening is cancer. Yet anybody listening for a few seconds will say, well, there's a slight flaw in that logic. Because if you're really cracking all of these codes, surely these codes matter in things outside of cancer. I mean, the cell, no matter how complex it is, there can only be so many mechanisms that go crazy. And you would be absolutely right. So let me close on 2 quick examples. These are photos representative of a disease that I must say I particularly hate, if you'll forgive the word. It's called myelofibrosis. And for some reason, if you can imagine a building site, the marrow gets filled with fibrotic tissue to the extent that it can no longer function. You've got genes, and they remember, okay, I remember in the womb I could make blood in the liver and the spleen. If I can no longer make blood in the long bones, let's retreat there. Except now you're an adult. So you do your best to make blood in the liver and spleen. They have very painful limiting capsules when you expand them. And we used to do clever things like radiate the spleen. When you take out or radiate the spleen, it goes to your liver and then you die. A pretty miserable, painful, inexorable way to go. This will be much more familiar to you. Vicious advanced rheumatoid arthritis. Why am I showing those 2 photos? Because they've got a target in common. They have a family of kinases, enzymes, just like the ABL I mentioned in CML, that are critical to driving them. And now we have the privilege of working across the old boundaries. What you learn in a patient with rheumatoid arthritis may be pertinent to somebody with myelofibrosis and vice versa. And so in closing, I mentioned CML earlier, and a lot of people said, oh, you were just lucky with CML. That's not going to happen again. it was simple. And we all knew that was rubbish. We used other words, but I think you know what I mean. But you had to wait for, proof. Everything in medicine is proof. Well, Steve's company have the lead drug that targets BTK, Bruton's tyrosine kinase. And guess what? If you change the M mutal, chronic lymphocytic leukemia, the commonest leukemia, this drug is the magic pill for that disorder. It can do things that we've never seen before, and it's only the start. Now, the reason I put plus, plus, plus is if I've stayed anywhere close to time, I was fascinated to discover a few weeks ago that every 3 minutes, a person under 18 years of age is admitted to the emergency room in the United States with a food allergy. Every 3 minutes. And every 6 minutes, half the time, it's anaphylaxis. It's life-threatening. Guess what's a drug that we think might be pretty good for those poor kids whose life are at risk? This same target, this same drug. And that's one of the things we're looking at Northwestern. So what I'd like to convey is not alone have we concrete room for enormous progress in cancer. That's due to real science, real collaboration, and what's more, all of the advantages are not going to be confined to cancer. Thank you very much. We don't have our routine with the microphone worked out like Mary, so let's just hand it over.
Okay, thank you. Very good, thank you. And I'm wondering if you can hear me without the standing mic. I think so. So just to begin with, I want to thank the organizers for the invitation this evening, particularly Alan and C2ST and Mary. For the opportunity to talk this evening, as well as Frank, again, for the definitive view of how we treat cancer today. You should know, Frank, that I got a text while you were talking from 60 Minutes. They want us to come and do 60 Minutes routine this Sunday based on what we did last night for Chicago Tonight. So. My objective over the next 20 minutes or so is to provide you with a view of what the kinds of cancer medicines that we might be using in the years to come. And so actually the talk is not how we treat cancer, but how we will treat cancer. An overview, an outline of what I'm talking about tonight is shown here. I'll begin by talking about some of the unifying characteristics or hallmarks of cancer. I'll then move into 3 examples of new therapies. Number 1, the idea of targeting cell survival. Secondly, the idea of controlling drug delivery by way of antibody-drug conjugates. And finally, I'll finish by way of a description of the whole idea of modulating of the immune system in order to allow it to attack tumor cells. And then at the very end, I just want to add a couple of editorial comments on what I think are important ingredients for us to be successful in coming up with new cancer drugs in the future. In order to know where we go next, you actually have to know something about how we got to where we are today. And so what I show here is that, again, cancer has been historically treated by way of surgery and radiation and nonspecific chemotherapy, some examples of which are shown here. You see the advent of more selective agents in the form of hormone therapy beginning in the 1970s. Then you begin to see targeted agents, those agents that are specifically designed to distinguish between normal cells and tumor cells. They start appearing in about the turn of the century. And at the very bottom, you can see the advent of cancer immunotherapy in the last maybe 5 years or so. And this is something that I'll come back to in a moment. 2 points about this slide. Number 1, while some of these specific agents have come and gone, it's important to recognize that all of these general categories continue to be very important for the treatment of cancer and will be for the foreseeable future. Secondly, I think there's the whole aspect of perspective. If you put yourself on that star and look backwards 20 years, it's easier for us to recognize how little we knew back in the 80s and 90s. Yet it is also absolutely guaranteed that someone speaking at this meeting 20 years from now will say the very, very same thing about us. So that progression of new anti-cancer agents has paralleled a very dramatic increase in our understanding of the biological processes that drive malignant growth. Again, cancer is a very heterogeneous set of diseases ranging from leukemias and lymphomas to various solid tumors. Yet despite that vast degree of heterogeneity, there are a common set of characteristics or hallmarks that cancer cells acquire that collectively characterize malignant growth. An initial list of those characteristics, those hallmarks, was published back in 2000 and included, for example, the ability of tumor cells to grow without instructions to do so. It included, for example, the ability of tumor cells to invade and to metastasize into new tissues. It included, for example, the idea of cancer cells to sequester new blood vessels and sustain angiogenesis. While that first list provided kind of some unifying principles on how is it that cancer cells grow, it was clear at the time that that was inadequate to fully characterize cancer in its fullest. And consequently, there was an expanded list of hallmarks that was published in 2011. That included, for example, the ability of cancer cells to cope with mutations and an unstable genome. It included, for example, as Frank mentioned, the different energy requirements of tumor cells versus normal cells. One of the characteristics that appeared on both lists relates to the whole idea of resisting cell death or evading apoptosis. And this is an area that we got interested in many years ago, as well as other researchers. Apoptosis refers to programmed cell death. It's the means by which organisms dispose of damaged or unneeded cells. It is characterized by a very highly orchestrated set of molecular events that leads to DNA fragmentation. It leads to the cleavage of intracellular components and ultimately changes to the surface of cells that allow them to be cleared without inducing an inflammatory response. Regulated apoptosis is important in any number of physiological processes. For example, the cell turnover that's necessary in embryo development, where fingers and toes are actually formed by way of apoptosis of cells between the digits. Dysregulated apoptosis, either too much or too little, can drive human diseases. For example, inadequate culling of white blood cells can contribute to autoimmune diseases like lupus. Dysregulated apoptosis obviously has an impact on cancer as well. Cancer cells have lost the ability to know when and how to die properly. And that can contribute to tumor initiation. It can contribute to the progression of already established tumors. It can contribute to resistance of tumors to therapy, all by way of allowing tumor cells to avoid some of the signals that normal cells would otherwise have to say die. In the 1990s, it became evidence that there were two pathways that regulate apoptosis in mammalian cells. Number one, an extrinsic apoptosis pathway, where the cells receive signals from the environment and interact with something called death receptors that trigger apoptosis. And then an intrinsic apoptosis pathway, which is mediated by a family of proteins called the Bcl2 family. Both of those pathways converge on the activation of proteolytic enzymes called caspases that ultimately begin cleaving cell components in a very orderly manner. The mechanism by which these Bcl2 family members regulate apoptosis is shown on this slide, and it is absolutely fascinating. It turns out that some tumor cells, the fate of some tumor cells is governed by the balance between pro and anti-apoptotic Bcl2 family members. Some of those family members are shown here and include the BH3 proteins. Those proteins are responsible for sensing cell damage. When they do, they bind to two other Bcl2 family members, BAC and BACs. That induces those BAC and BACs to form oligomers and eventually form pores in the outer membrane of mitochondria. And that allows cytochrome C to be released. That causes the activation of caspases, and that causes cell death. The anti-apoptotic Bcl2 family members hold this process in check. They bind to the BH3 only proteins and inhibit this entire process. So the idea that when we got started in this whole area was, could you come up with drugs that bind to these anti-apoptotic Bcl2 family members, consequently liberating the pro-apoptotic Bcl2 family members to trigger this process in tumor cells? So that was the concept. We started this 20 years ago. And the way we started is by better understanding the interaction between the pro-apoptotic family members, shown here in green, and the anti-apoptotic family members, shown here in orange. With a better understanding of how those two fit together, then we developed an entirely new drug design process called SAR by NMR that provided us, that gave rise to molecules with sufficient affinity to bind to the orange protein, release the green protein to trigger apoptosis. That ultimately led to, in 2003, to a compound 737, which reliably induced apoptosis in tumor cells. The problem with this compound was that it was not orally available, and we proved to ourselves that we needed chronic therapy. And so we went back into the lab and ultimately reworked the structure of 737 to give rise to a compound called navitoclax. And that was the first Bcl2 inhibitor to be dosed in a cancer patient. The activity of navitoclax in those cancer patients was fascinating. We did indeed see anti-tumor activity in lymphocytic leukemias, but we also saw a precipitous drop in circulating platelets. And that turns out to be a consequence of the fact that navitoclax inhibits multiple Bcl2 family members. It inhibits Bcl2 and Bcl-XL. And that Bcl-XL activity drives the survival of platelets. So we had to go back into the lab once again and rework the structure of the molecule such that it only inhibits Bcl2. And that eventually gave rise to a compound called venetoclax, which now has shown very, very striking activity in chronic lymphocytic leukemia patients and is being vetted by the FDA for approval. This compound also avoids that drop in platelets in cancer patients. I should mention that this particular slide makes the discovery folks at AbbVie cry, because we have 20 years of blood, sweat, and tears on one single slide. So, venetoclax provides one opportunity to target one of the hallmarks of cancer, again, resisting cell death, apoptosis. And again, that's something that has not been available to cancer patients heretofore. The field has made headway on targeting other hallmarks of cancer in the meantime. Some of these are shown here. Some of those are small molecules, small drug-like molecules, akin to venetoclax on the left. Other of these molecules are antibodies that very specifically target proteins on the surface of tumor cells. While these small drug-like molecules are very potent at killing tumor cells, they also distribute broadly through the body. and consequently can have unintended side effects. Antibodies, on the other hand, have a capability of very, very nicely targeting tumor cells. So one idea that has been around for a long time is the idea of trying to control the delivery of these molecules by actually attaching them to antibodies, giving rise to the compound in the bottom. And that's the whole area of antibody-drug conjugates, ADCs. What we're doing here is to take those toxins and actually hook them to the antibody. The concept is pretty simple here. It is systemic delivery of these antibody-drug conjugates allows those conjugates to hone or localize specifically to cancer cells that have the intended target. Once the antibody-drug conjugate sees the intended target here in purple, it's internalized and the payload is released where it can kill that cell, but not cells that don't have that purple target. It took, just like in the field of apoptosis, it took a long time to make progress in the field of antibody-drug conjugates and eventually to benefit cancer patients. And that's because of some very specific requirements that are needed to get these antibody-drug conjugates to work. One of those requirements is the need for some very, very potent anti-cancer agents. Two of those are shown here, the dolastatins and the maytansinoids, and both of these kill tumor cells by inhibiting cell division. Interestingly, both of these come from natural products. The dolastatins from a sea hare in the Indian Ocean, the maytansines from bacteria associated with the roots of plants in South Africa. Another one of the very definitive requirements for antibody-drug conjugates is that these antibodies have to localize very, very specifically to tumor cells. You don't want to have the payload being delivered to normal cells. And so one such antibody is something called ABT806, which targets a protein on the surface of cells called EGFR. What I show here on the bottom left, what we've done here is to implant tumors into the flanks of mice. And then we have dosed a version of ABT806 that you can track its location by way of imaging. On the right-hand side, you can see that 806 localizes very, very nicely to tumors that have high EGFR expression. But on the left-hand side, you can see that those tumors don't have high EGFR expression, and consequently, there's no localization of the antibody. Similarly, on the right, you can see here images from a cancer patient with a brain metastasis, and that ABT806 nicely localizes to that lesion. What we've done now is to take ABT806 and adorn it with one of those dolastatin analogs to come up with an antibody-drug conjugate called ABT414, which is now being studied for a very, very aggressive form of brain cancer. Two antibody-drug conjugates have been approved so far. One is Adcetris for the treatment of Hodgkin's lymphoma. One is Kadcyla for the treatment of HER2-negative or HER2-positive breast cancer. Adcetris uses the dolastatin payload. Kadcyla uses the maytansine payload. Interestingly, as you can see, we like to use acronyms ADC, and you can find ADC in Adcetris, and you can find ADC in Kadcyla as well. So one of the hallmarks that was left behind in the original list was this whole idea of avoiding immune destruction, and the whole idea of using the immune system to attack tumors. This is a field that has blossomed a good deal over the last 15, 20 years, and now some of the breakthroughs that we've seen in immuno-oncology are on the surface of, or on the covers of Science and Nature, as well as Newsweek, as well as Time Magazine. It was not always that way. If you went to a cancer conference 15 years ago and wanted to learn something about immuno-oncology, you would have to get up early and go to very small remote classrooms in order to see those sessions. Nowadays, you still have to get up early, but that's only because it's a standing room only crowd at some of these major conferences. This is a field that has absolutely burgeoned over the last couple of years. Again, nature taught us more than 100 years ago that indeed the immune system could actually engage and control tumors. There was a surgeon in New York named William Coley who noticed regressions of tumors in sarcoma patients sometimes coincided with those patients having post-operative skin bacterial infections and the corresponding high fever and the corresponding inference of an immune reaction to that targets the tumors. William Coley goes on to try to treat experiments with the idea of trying to treat patients with heat-killed bacteria. And in fact, he was able to show some durable responses in some of those patients. But the field essentially goes dormant for many years because we really had no clue about what was actually stimulating an immune response in those patients and actually how that all worked. Advances in the field of immunology helped us begin to understand what we could do here and gave us a better understanding of how is it that cytotoxic immune cells could actually then recognize and take out cells that are deemed normal. And now we know that actually the immune system is in constant battle with tumor cells, and actually those tumors progress when mechanisms of immune surveillance are lost. So this slide just shows very briefly how is it the immune system can control cancer. The dendritic cells must first take up tumor antigens. They must make their way to lymph nodes where they then need to engage T cells, cytotoxic T lymphocytes. That needs to activate those T lymphocytes. Those T lymphocytes need to make their way back to the tumors where they need to become sufficiently activated in order to kill tumors. One thing that's pretty obvious is that tumors use multiple mechanisms to avoid destruction by the immune system. And one of the ways they do that is by way of T cell checkpoints. It turns out that those T cells can indeed recognize tumor cells as foreign, but the activation of those T cells is held in check by something called co-receptors, which bind to ligands on the tumor cells and essentially dampen the response of those immune cells. It shuts them off. The big advance that's been made over the last, say, 5 years, where we've seen some very, very interesting and dramatic activity in various tumor types, is the fact that molecules that disrupt this interaction, that is T cell checkpoint inhibitors, essentially release the brakes on these T cells and allow them to re-engage the fight against tumors. And now we have multiple different agents that have been approved in, as Frank mentioned, in the area of melanoma, in the area of several types of lung cancer, in the area of kidney cancer. Another one of the more transformational types of agents that's being looked at in the clinic right now relates to chimeric antigen receptors. This is not really a drug in the classical sense. It's not a drug or an antibody, but rather a living drug, because what we're doing here is treating cancer patients with cells. What we do here is take blood from cancer patients, we extract the T cells from that blood, and then we modify those T cells to express a receptor that specifically targets tumor cells. We then grow those chimeric antigen receptors in the lab and then reinfuse those cells into patients after they've undergone chemotherapy in order to strip away any residual tumor cells. Again, some very interesting activity of these agents in certain acute leukemia patients, both adult and pediatric. patients. So that gives you a view of three different new types of cancer therapy that are on the horizon. Again, the idea of targeting apoptosis, the idea of antibody-drug conjugates, trying to target the immune system. I just want to close with a couple of comments on what I think is absolutely essential for us to make headway going forward in this area. And the first relates to top-notch scientists. In order for us to succeed going forward, we are going to need the next generation of scientists to get interested in this field. And that, what I show here are all the scientists that have contributed to the programs that I've talked to you about at AbbVie. They're all smart, they're all dedicated, they're all hardworking, but none of them were born at AbbVie. Rather, they got interested in science and they got interested in oncology science somewhere else. And that's why the role of C2ST and the mission of trying to enhance the science acumen within Chicago and beyond Chicago is so important to all of us. The other piece I think that is absolutely essential for us to be successful going forward is the whole idea of collaboration. None of the breakthroughs that I mentioned here this evening would have occurred in isolation. Rather, they occurred by way of interaction with scientific luminaries like Dr. Giles and Dr. Hendricks, scientists in the pharmaceutical industry, scientists in biotech, consortia, all working together for a common cause. And I'm sure you recognize the lorry on the very top of this slide here. And with that, I would like to thank you again for your attention.
Well, thank you very much for that very stimulating presentation, both Dr. Giles, Dr. Davidsen. We're having a little bit of a malfunction with the microphone. Can everyone hear me in the back? I'll be sure and use it. Okay, thank you. I'm going to invite questions from the audience. Don't be shy, especially our students who are here. Well, yes, sir. You're first.
I have two questions which I think are related. You talk about collaboration, tearing down the research silos. President Obama particularly talked about that. How, in fact, is that organized? How does this collaboration happen? And what kind of improvements do you see in that in the near future? And secondly, why didn't you guys talk about CRISPR? You talk about immunology, but there's a whole other pathway out there. And why didn't you talk about it?
I'll handle this. I'll handle the CRISPR piece. So the gene editing piece is very much on our minds. That whole idea of transforming those T cells that you get from patients, we can use CRISPR technology to begin to edit the genes from those T cells to make them even more active than just adding the receptor. You're exactly, and so CRISPR is, you should know, is the very, very interesting new way of editing genes very, very selectively, and I think you'll see that technology come to fore within several years. But the example I could cite was the very last one, where we're going to use patients' own T cells to modulate the tumors. I just want to mention one part of the collaboration. You wanted an example. The example I have would be the fact that a team of scientists at AbbVie is working with Mary, who has a very, very interesting idea on a protein called nodal, and we are working together to try to see how does it work, where does it work, what tumors might be sensitive, and so on.
I didn't think we were allowed to talk about it. No, just kidding. So I'll just tell you how this came about, because you talk about how you catalyze these types of interactions. Dr. Giles was hired by the Lurie Comprehensive Cancer Center, and working with Dr. Platanius, he said, hey, I have a great idea. Let's have a half-day session or all-day sessions dedicated to exchanging scientific information with local, regional, and national pharmaceutical companies. And AbbVie was one of the first companies who was invited to the table. Dr. Giles invited me to present some of our interesting scientific findings from the lab. And this was the greatest opportunity at all, to be able to present, to have these incredible questions asked, and then to spark a collaboration that has now gone on for over half a year. And it's so important, and I would just say, especially to the students in the audience, scientists have to publish, publish or perish, but it's very important that other laboratories and other scientists are able to repeat your data and your observations. That's the importance of conducting research in full transparency of the public. But it's also important to have this opportunity to work with a pharmaceutical company. They have to be able to repeat our data and then take it to the next level. So that's how this started. And Frank, thank you again for getting us together. Okay, other questions? Yes, sir.
I have a very personal, specific question, but I think would be universal, because I think most of the people here probably know someone who was battling cancer. Well, I have an Army friend from Army days. He's about 73. He's battling cancer down in Naples, Florida. How can I, in a personal way, be helpful to him? Do I ask him, are you getting the best kind of therapies down in Naples, Florida? Do you know anything else about other therapies like we're doing up at the Lurie Center? How, in a way, would I approach them in this question? Like, would your Department of Hematology or Cancer at Lurie review his papers and see what's being done, or do they have to see him in person?
So great question, Frank.
So maybe actually
I
don't.
think we need to repeat that.
Okay, let me repeat that, and I'll just summarize briefly. This gentleman has a friend who's in Naples, Florida, who has cancer, and so the question is he receiving the right treatment, and how would we know how to help him if he was or was not receiving that treatment? So we're going to direct that to Dr. Giles.
So I think, Mary, actually both questions have one critical part of the answer in common, and you alluded to it, and that's communication. I still remember, I don't know, X years ago, which I'm not going to specify, starting in a new job, my first job in the United States. People there were extremely enthusiastic about a new study that we're just about to launch. And I'm in the back of the room debating, I'm not sure this is how I want to start my first job, but somebody has to do it. And I stood up and said, well, we actually tried that in London last year and it didn't work. The difference is the web and technology, because that would not happen now. And the short answer is, anybody who wants to know about nodal, N-O-D-A-L, can go in, find everything that's being published, find how much of it has been replicated by whom and when. And the answer, which is really one of the most satisfying parts of our daily life, is in the middle of seeing the patients you're physically seeing, there are all these virtual patients. A simple question. I'm getting this. And of course you try and be courteous. You're not going to try and make on-the-fly comments without seeing somebody. But what you're referring to is telemedicine, the sharing of opinions, what are the options. And that has increased, the communication is incredible. It's the critical word. And as Mary said, it's accelerating the process every day, because of course there's a thrill in finding something first. That's the nature of what we all do, and it's very exciting. But the real thrill is when somebody else replicates it and said what Mary said was true. That's become part of the discipline. Except now it's all in the public domain. And then you have, our partners in industry who have to apply a certain degree of rigor and say, well, is it true, but is it commercially viable? Will it get regulatory approval? These are the steps. So now all of these things run in parallel. As nodal is being developed, and for what it's worth, I'm certain it's going to be a mediator of an effective therapeutic approach. But now you have diagnostic approaches that go with each new thing to say, well, instead of giving it to 100 people and hoping it will work in 10, a priori we'll try and define a companion diagnostic as who are the 10. So all of it is running in parallel. All of it is instantly communicated. For example, to give you a very practical point about this region, there's immense collaboration between the Robert H. Leary and the University of Chicago Comprehensive Cancer Centers. And in this city, we have 2 of what's less than 50 in the country. And of the 50 comprehensive cancer centers, not all of them see patients. And in the specific funding that the government gives now, the more we collaborate with sister comprehensive cancer centers, the better it is for everybody. So I would say the silos, they'll never fall fast enough. They could always fall faster. But the degree of communication is part of the answer, and it's going in the right direction, and we'll be very happy to talk to that person or talk to them in an appropriate, de-identified manner and say, well, this is what's available, or these are things, or refer them to a sister institution down there like the Moffitt, who are in this comprehensive cancer centre network. It's all communication, and it does happen.
Oh, you have a question? Yes, fellow board member.
This is a wonderful program, I must say. And it's extremely important to communicate, as you say, the information. Because another problem that arises as this new event, a new discovery comes, but in a clinic, then you have the industry that pay the bill, that refuse to pay for certain events. So I think it's extremely important that we try, go on the 60 minutes, please, go it twice, because we need to have some of this stuff available to the public, not to some elite only.
Yeah, but I think we, if there's anybody here from Blue Cross, I'm going to regret this, but I don't care. I think we need to be very, very careful about the word industry. Part of the Affordable Care Act is to drive home something that should never have had to be put into legislation in a democracy. And you know what that is? That is that many of the health insurance companies, and the ones specifically in Illinois, specifically lobbied to do this. And that is that if you're the payments for standard of care tests, that they would be legally obliged to pay for you. If you go on a study, which increasingly might save your life or prolong it, they specifically excluded what is in common between the closely allied political states of California and Texas. In those states, an insurance company cannot deny you standard of care tests. They must deliver the same standard of care test whether you're on a study or not. In this state, that was lobbied against and effectively surgically removed for early phase studies. So we need to be careful because frankly, the pharma industry does pay their research costs. Reliably, constructively, and immediately. They're not the industry that, if we want to talk about reimbursement, that's retarding these developments. So if there is anyone from any of our esteemed insurance for-profit companies, they may feel free to defend or address that statement. It just so happens to be true.
That's a good comment. Yes, sir.
This isn't directly relating to cancer, but you mentioned that individuals can find articles on their particular disease and follow the literature. But in reality, when you go to certain publications, there is a $35 download fee or a fee for just an abstract of what information they're trying to obtain. Is there any means by which there is a site where individuals could have access to that information?
So,
Universities do. I'm privileged, I can get access to most publications for free, but the general public doesn't have that privilege. The second question I have is, how do you convince the NIH to pay to replicate a study?
well, there are two. First thing is the great majority of the journals that I think most of us would have respect for, they preserve a certain kind of media lead of 6 months or a year, and after that, many of the key articles are generally available. So there is a lead time bias, but they have their own costs, and that's how they justify it. It's fine by me. The second is the NCI and the NIH. Maybe Leon or Mary would care to comment on that. We heard earlier about the moonshot, and
Buried in the small print of the moonshot fascinates me that some of the moonshot tax money will be spent to help fix the FDA. So I'm thinking that's great. So we're going to spend more taxpayer dollars funding the taxpayer dollars that don't work right now. That doesn't sound particularly scientific. And in point of fact, there should be a lot more emphasis on, you know, the NIH does phenomenal work. The Comprehensive Cancer Center Network has done more to advance anti-cancer therapy than any funded entity on earth. But there should be more scrutiny of exactly where all the dollars are going. And I think it's all going in the right direction. Collaboration has been encouraged, practically. There is an effort to increase the funding. It's getting harder and harder to become a comprehensive center. It's getting harder and harder to stay that way, which is exactly the way it should be. Funding is always an issue, but I think the funding here, anybody who thinks the funding here is bad might try and look at Europe and see how much is coming out of there.
No, so it's a good point. And I would just add to that point that I think the best watchdogs in this field for all of us happens to be our colleagues. You know, an exciting observation comes up, and believe me, other individuals who are in that space focused on that topic are going to try to repeat the data. But I'm just going to add one other variable, parameter, that we really don't talk about very much, and that is the uneven quality of reagents that are commercially available. I have tried to offer an article, a publication on this to several different journals, and they will not touch this subject because in so doing you have to implicate certain commercial entities that are providing antibodies that are not good, have impure products. And so this is kind of a dirty little secret in our business, and it's one that I think we'd really like to bring out a little bit more so others will be aware of it. It's part of the problem that contributes to this irreproducibility of data. So I would mention that. Yes?
And about the access to the scientific articles, there is a ResearchGate website, which is equivalent of like LinkedIn for scientists.
Okay.
You can make a profile and you can directly request a scientist for this specific publication if you want, if you don't want to pay.
You know, that's a great point, and I'm going to ask the C2ST staff if they would be willing to offer this advice at the website, how to access information. Oh, were you able to hear? Okay. I'll talk with, oh, you mean to hear the question? You didn't? Okay, so the response was that there is a mechanism for accessing via ResearchGate publications. And I said, you know, this is C2ST. We can help. We'll put this information on the website and promote it. Thank you. Yes.
Could I add a follow-up to my original questions? And that is, if someone wants to do research on the web, I strongly recommend starting with Wikipedia to get terminology so that you can start searching. And then go to PubMed and look for things called mesh terms, M-E-S-H. And if you can get those right, you can find articles much easier than if you just put in keywords and hope.
Everybody hear that? Don't put keywords in and just hope. Okay, there's a mechanism. I'm going to have this as the last question from the audience. Yes.
I don't know if this is accurate, so I'd like to ask, but I've heard recently that anybody diagnosed with cancer now, they do a complete genomic sequencing on them. And I'm wondering if that's only happening at major research hospitals or if it's going to be mandated somehow. Because to my knowledge, I haven't gone through that. So, you know, how does one get that? I know even testing for specific genes associated with your type of cancer, in many cases you can't get doctors, probably insurance companies, to approve that, you know, because of the high costs. And there's a lot of resistance and pushback when you ask them to do any kind of this kind of testing. So how do you change that?
Good question.
So I'll take the science piece and then turn it over to Frank for the practical piece. But in terms of whole genome sequencing, yes, that capability exists. And you can do it in a bunch of different ways. You can assess the sequence of the whole the whole genome. You can assess the sequence of just those genes that are transcribed. You can do it in just genes that you know are transformed in cancer. So there are different levels from a menu. There are different levels that you could do to assess what the genetic abnormalities are for a given sample. How that is reflected practically, Frank can help you with that.
Well, I can give an answer, but I'm not sure it's helpful because I wish that what your opening remark was anywhere close to the truth. The fact of the matter is it's a tiny, tiny minority of the patients who should have molecular diagnostics performed. I'd say that the Lurie is one of them, frankly, most focused on this, as focused as anywhere on Earth, and specifically and more importantly doing something with the information. And quite honestly, you know, about 18 months ago, we had to make a choice, and that was, is it our job to try and spend all day long fighting with the insurance companies and fighting with everybody, or are we actually going to take care of the people in front of us? Because somebody asked me earlier, you know, what are the resources and what are the money you need to accelerate this? A very sad fact about is to have two pictures that are simultaneously visible. This is exactly what I could do with the information, and this is where it would lead. And these are the tiny numbers of patients that I can even run the tests on. It's an incredible complex matrix. In other words, you have whole sets of companies who say, you know that lung cancer slide, for example, I showed you? Well, I can offer you a cost-effective test, and I'll test for 4 or 6 or 8 or 10 of them. And you're looking at them thinking, excuse me, I want to run the whole sequence. Because how did we get from 11 to 12? How do we get from 12 to 13 to 14 to 15? So you have people trying to kind of keep selling you hard, I'll do the tests you know about, and you're saying, no, 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 what I'm interested, I know about the ones I know about. The next patient may have one of the ones I don't know about. So you have this battle where we're trying to keep it wide open and get as much information that's potentially actionable as possible. And you have the insurance companies trying to pay for nothing. And then you have an industry in the diagnostic side fractionated from, I'd like to move to whole exome as quickly as possible. And another section trying to say, well, I'll do 2 or 3 tests and I'll sell those to your department of pathology and we're all happy. I wish that your opening remark was anywhere close. You know, we would like and are striving. The OncoSet program, it's part of our unremitting drive, is to be able to have everybody have at least a reasonable facsimile of a decent molecular screen. We're nowhere near it, and we're one of the most focused sites on Earth. The economics of that, you know, as a society, people are going to have to me, to have that technology available and for the benefits to be no longer ephemeral, we have FDA-approved drugs based on this knowledge, and to have a rate-limiting step in this country that you can't even generate the basic information, that's something society has to address because it's unacceptable.
Steve.
I just wanted to amplify on that because it's a very important point. Frank mentioned actionable. That is, you can read the genetic abnormalities, and then you can look at them and say, here's one that we have a drug that could address. That's actionable. You can pick the drug and treat the patient based on whatever lesion you see. For many, we do not yet have actionable, we don't have the drugs that would fix the problem. That's where we come in. And Frank circled all kinds of new targets. We're trying our best to come up with the next agent that could then be actionable for a particular genetic lesion, so that when people look through their genomes from a tumor sample and say, yep, we have something to give you. Right now, that's the exception. Right now, we look at this and hope that there's one that matches. Oftentimes, they're not. But that's why we're in the business. And I just one last thing, and that is the young guys out here. It's that last slide that I had up there is absolutely true. If you have any interest in science, it's cool. And it's an absolutely fun job to get into the world of oncology science. it's benefiting people ultimately. It's extraordinarily satisfying, and the science is amazing.
That's our pitch.
Two questions. How important is big data in what you guys are doing, and also the moonshot thing? You know, it is going to be run by the idiots in Washington. So how do you really feel about that?
Okay, very last question, and I'll just repeat it. What do you think about big data? And what do you really think about the moonshot program, because the government is going to be involved? Okay, who wants to take this quickly? Steve?
I'll do the big data. He can have the hard.
No. Okay. Neither of us will address the latter question.
Look, the big data is just what we just talked about. That is, you can do the sequencing across all 23,000 genes in the human genome. And you can generate tons of data from a single sample. The question and the hard part for all of us is, what do you do with it? When you truck through all of it, A, C, T, G, and whatever it is, then what do you actually read from it and decide from it? And so while the analysis of the big data is a key, it's actually transforming that into something that you do is the tricky part.
And to amplify on that, superimpose that image on the iceberg. So now we have data on the code, like reading the code. But then the code has to make a protein. Well, does it or doesn't it? Then there are other proteins that will move in to stop that protein doing its job. Or after the protein is manufactured, as you know, it may be epigenetically or otherwise modified. So it's a challenge, an enormous challenge to handle the data we have. We're beginning to get a glimpse of how much data it's going to take. But really the point is, that's the challenging side. Look at the other side. It already is resulting in drugs that are working where no drug ever worked before. So yes, on a bad day, like we all have, you're intimidated. And on a good day, you say, well, if we know this much and we've achieved so much already, what's going to happen when we have the rest? But we're absolutely dependent on the full spectrum of science now. We're investing to the hilt in bioinformatics, in what's a safe cloud way to store this data. All of that, all of science is involved in this endeavor, and it's not just pertinent to cancer, because you have to look at the other end of a logical sequence. There are only so many aberrations that are possible. That's a fact. There are only so many that are possible. Logically, those aberrations with twists may give one person rheumatoid arthritis or the other myelofibrosis. So sooner or later, you're going to reach a threshold where the benefits will ripple exponentially. And data and the incredibly clever people who develop the algorithms and tell us, out of all of this noise, that's where you need to look. And then just think of it as a cascade of marbles. Like you might have 15 actionable. But if you find the magic one that pulls out the base, that may buy you 2 or 3 years with that tumor. That's how we think. You keep adding it up until the whole structure falls apart, the apoptosis kicks in. You know, you can easily see, let's say the cell is refusing to commit suicide like a normal cell. You tell it, commit suicide. Oh, and by the way, to help you, this T cell is keeping an eye on you, and if you don't, it'll kill you anyway. They are utterly, you know, these are whole new waves of getting the cancer out of there, and you can see why the optimism is logical. These are non-overlapping or minimally overlapping mechanisms of action. That's why they work, and we have each of these are moving in parallel. In the next few years, you're beginning to see them intersect.
Well, please join me in thanking our wonderful speakers.